So welcome to Micro Seminar, everyone. We are very excited to have Siobhan Watkins doing our talk today. She had her PhD from the University of Portsmouth, did a short postdoc there, and then moved to Loyola University for a postdoc. And she is headed, or already in New Mexico, but headed down to New Mexico Tech to start a faculty position this fall. So congratulations on that, Siv. And she is extremely entertaining and has promised to try to fit the entire bingo card of slang in there, so we'll see what she gets through. But with that, we will turn it over to Siv. So if you want to share your screen and get started, we will have questions at the end of the seminar. You can tweet them to MicroSeminar or use the hashtag #USeminar to ask any questions, or if you're in the Hangout, check in then and we'll get questions from you. So take it away, Siv.
Okay, did that work?
Yep, looks great.
Okay, I'm going to switch my big screen off so I don't get distracted. Okay, so first of all, thank you very much for inviting me to take part in this. This is, it's really cool, and I'm really happy to be doing a presentation today. I'm in quite illustrious company with some of the other people who have given a talk in this series. It's a little bit intimidating, so I'm going to start off with a disclaimer, which is how all of the best seminars start, innit? I'm a very newly minted assistant professor at Tech. I only moved to New Mexico a few weeks ago. I'm not even really properly installed at Tech. I'm in Los Alamos for a little while, and so I haven't really done anything. But luckily, at 1 or 2 points in the last 10 years, I have been productive a little bit. So I'm going to talk about some of the stuff that I've done in the past and do a little bit of cheerleading for what's going on at Tech and at the New Mexico Consortium. So because this presentation is kind of cobbled together a little bit from other presentations that I've done in the past, the aim here is really to have made it as accessible as possible. So many apologies if you're watching this on YouTube or whatever and thinking that I've pitched it at a really stupid level, at a really low level, and that I've completely buggered it up. I'm sorry, I haven't written it for you. I've written it for undergraduates, and hopefully this is going to get some younger students interested in what I'm doing. So that's my disclaimer. What's with the stupid title? Most of the work that I'm going to talk about now was done at Loyola University Chicago in Catherine Patonti's lab. That's me and Catherine there. We were just about to order some deep fried cheese curds, which we subsequently dipped in fondue. So we were really living the dream that day. And by training, I'm a bench microbiologist. I'm probably what you'd call a traditional microbiologist, whatever that means. And Catherine's a computational scientist. So this is a really nice mix. We had quite a nice trade-off. We spent a lot of time saying this to each other. There were a lot of 3 AM conversations via Google Chat with one of us saying, have you completely lost your mind? That's not what this data is showing us. So that's a good way to do science, right? And similarly, 99% of the work that came out of the postdoc at Loyola was driven by undergraduates. There's no PhD program in the biology department at Loyola, and all of the people on this slide, aside from Jason, who's a postdoc, and a handful of grad students, they're all undergraduates. And these guys were all really intuitive, really creative scientists, and they were able to come to me and tell me that I was wrong a lot, which is brilliant, and then I told them that they were wrong, and then we had a big argument, and then we did some more work and figured it all out, and I was really happy in this lab. This was a great place to work, but again, we spent a lot of time saying this to each other, and I think this is a really good way of doing science generally, is having this constant discussion and a constant kind of critical analysis of what you're up to. So that's where the title of the talk comes from. Maybe it makes a bit more sense now. So aside from babbling incoherently for the first few minutes of a presentation, I like to have a quote at the beginning from somebody who's a lot smarter than me. And this is from Tom Curtis, and he quite famously said, "I make no apologies for putting microorganisms on a pedestal above all other living things. For if the last blue whale choked to death on the last panda, it would be disastrous but not the end of the world. But if we accidentally poison the last two species of ammonia oxidizing bacteria, that would be another matter. It could be happening now and we wouldn't even know." When you put it that poetically, it's kind of hard to argue with. And microbiologists have known for ages that bacteria have a critical impact on the processes that allow for life on this planet as we know it. But what Professor Curtis didn't mention in this quote, and why would he, is that bacteria themselves are subject to considerable external pressures, and not least from these guys. So these are bacterial viruses, viruses that infect bacteria, bacteriophages. This is what I study. This is what I'm interested in. I'm a phage biologist. These are two viruses that we isolated from Lake Michigan a couple of years ago. And when you work with the kind of students that I've been privileged enough to work with, these viruses end up with names like this. So this is Veda and Bitey. Veda's going to pop up a little bit later on in the talk. But for now, I want to put this all in a bit of context. So I want to lay some numbers on you because everybody loves facts and figures, don't they? Yeah. So current estimate is that there are 10 to the 31 viruses in the biosphere. So Mum, if you're watching this later, that's a lot. And I'll try and put it a little bit more romantically. That's 100 million times more viruses than stars in the universe. In your face, physicists. I'm in Los Alamos at the moment and surrounded by physicists, and that's obviously had a weird effect on me. Anyway, these things are mind-boggling. They're tiny little scraps of genomic information floating around the planet in these really vulnerable little protein jackets, and they have a gargantuan impact on the planet, and it's really because there are so many of them. So in a litre of seawater, you're expecting to find 1 million microbes, and you can match that with 1 billion viruses. Right? We're not going to run out of any of these to study anytime soon. So that's me, looking kind of shiny and happy at an ASM conference. Mark Martin took that picture of me. Hi, Mark. And I like looking for these viruses in fresh waters and wastewaters. And I really sort of approach this from two themes. The first one being an exploration of diversity. What's there? What phages are there? And really, we don't know that much about that at this point. And I do that with lots of culture-based methods and metagenomics as well, and genomics. And I'm also really interested in host range. So that's, who are these phages infecting in terms of bacterial hosts? Because that has the potential to have a really big impact as well. And I'm a consummate fiddler. I'm never happier than when I'm in the lab messing about with bacterial viruses, or in a puddle or a pond somewhere, or a wastewater treatment system paddling around, or tweaking data or something like that. So I guess it's going to become pretty obvious over the course of this talk that I'm a big fan of tweaking and fiddling, and I think this opens up lots of unexpected avenues for science. So that kind of goes hand in hand with the I do not think it means what you think it means thing. So again, a little bit more context, and probably most of the people who are watching this will know all about this. My apologies again, this is for the benefit of my mother and undergraduates. Usually when we're talking about phage and bacterial interactions, we mention lifestyle. So phages have one of two main lifestyles that they can adopt. There's actually a number of subtle levels between the two, but these are the main ones. And the most obvious impact that a phage can have on a bacterium is by killing it, right? We call that lysis. So if we have a purple cell and a yellow phage, the phage starts off by adsorbing to the purple cell. It injects its genomic information into the bacterium, which is what this little yellow squiggle represents, hijacks the biochemistry of that cell, produces more viral offspring, the viral progeny are released, and you end up with a dead bacterial cell and lots more viruses than you started off with. And we can see this when you spread a bacterial lawn on an agar plate and apply viruses, the viruses will burn holes in the lawn. And we call these plaques, and they're sort of roughly analogous to bacterial colonies. We can count them, we can describe their morphology, and we use them to isolate, in theory, clonal cultures of phages. So what does this mean actually in the environment? Well, we take this sort of mock-up of a collection of bacteria community. We've got the purple cells that are dominant, the green guys who are hanging out but really not there that much because the purple cell is dominant. The yellow phage comes along, infects the purple cell, kills the purple cells, and this does one of two things. Because the death of these bacteria has released a lot of dissolved organics into that environmental matrix, some bacteria may show up that weren't there before, but also it allows for the green bacteria to proliferate because the purple cells aren't there anymore. So we've gone from this to this. And that's pretty good going for just one phage and one bacterium. When you start thinking about a phage that infects lots of different bacteria, or a really diverse community of bacteria and all of the associated phage that go with those cells, it becomes really complicated really quickly. And if you picture this happening on a massive scale every single day, you begin to understand where the impact comes from. So the second sort of main option that phages have for lifestyle is roughly equates to dormancy, and we call it lysogeny. Again, we've got the yellow phage infecting the purple cell. This time the purple cell contains a blue squiggle, which is a bacterial genome. The phage injects its genomic information into the cell, but this time an aspect of the phage genome is integrated into the bacterial genome. And at this point, we call that yellow squiggle a prophage. It's kind of a virus, but not a virus. And every time the bacterium replicates, the prophage replicates as well. And this can go on and on and on until maybe the life cycle switches back into lysis. So phages are really genetically promiscuous, and they're always picking up little packets of information and dropping them off in different places. And this obviously has huge implications for genetic mobility in the environment and everywhere. And for any lay people who might be watching this and interested, a really nice example of that is diphtheria. And the disease diphtheria is caused by a toxin that's produced by a bacterium, but the bacterium can only produce this toxin once it's been infected by a phage, which is carrying a really specific gene. It's really cool and has huge implications. So that's your context. Now I'm going to talk about some diversity studies that we did in Lake Michigan. This is the Lakeshore campus of Loyola University Chicago, right next to the lake. The hint's in the name. It's a pretty nice place to be in the summer. In the winter it's not so great, but you know, you deal with it. Why are we interested in Lake Michigan? Well, it's really big for a start. This is Lake Michigan superimposed over the little country that I'm from. I'm really sorry about everything that we've done in the last few weeks. Don't blame me, I voted to stay. And it represents drinking water for millions of people. It's really important in recreation. There are lots of concentrated areas of urbanization around the lake. So it's worth getting a handle on the general ecology of Lake Michigan at all levels. So for the whole time that I was at Loyola, we were constantly throwing undergraduates into the lake near the shore, collecting lots and lots of samples, lots of time series, lots of replicate samples. And very briefly, we would bring the water samples back to the lab and separate the water on the basis of size fractionation. So we were able to say, okay, we think this fraction contains all the bacteria. We think this fraction contains all of the viruses. At some point, there was a fraction that contained eukaryotes, but anything with more than one cell intimidates me, so I never really paid much attention to that part. And for the viruses, we take the appropriate fraction and extract all of the DNA from it. The total complement of DNA would be extracted from that water. And then we'd sequence the DNA. So for the purposes of the undergrads watching this, and my mother, when you do DNA sequencing like this, you start off with a huge big pile of raw information. And as time goes on and you process that information, you end up with less and less useful stuff. So you're getting rid of all of the junk, you're trimming away all of the fat, the stuff that's unhelpful or poor quality, and you go from a big pile to a small pile. So really, for the purposes of this presentation, all I need to tell you is that we started off with 15 million raw reads for 9 metaviromes. Everyone watching this probably knows all about BLAST. Again, this is just a really quick description for any lay people who may be watching. BLAST is a really nifty little tool which essentially compares your brand new DNA sequences to existing sequences in databases that we already know about. And it will give you a return of hits or matches and a rough approximation of the quality of those hits or matches, and that's on the basis of how identical your sequences are to the sequences in the database, how much your sequences cover the sequences in the database, and something called an e-value, which, you know, the lower the better. That's all we need to know. So for most large-scale DNA sequencing projects, BLAST-based classification has its limitations. This applies particularly to phages in the environment. For a start, our databases aren't very well populated, and that's really, we haven't been examining them in this way for as long as we have, say, bacteria. Which is why this kick for examining diversity is really important as well, because the more we can do that, the more we can populate our databases and use them for comparative purposes. Phages as entities on their own are, you know, we don't really know that much about them either. So when we have a genome of a new phage, the chances are we don't know what most of that genome does. So a lot of these genomes are annotated or labeled as containing hypothetical proteins, which in the grand scheme of things, when you're looking at phages, means sort of diddly squat. And hypothetical proteins is the bane of my existence, but we deal with them. And again, we have this issue with prophages. You know, the little yellow squiggle that was on top of the little blue squiggle. When you get matches to those guys, it suggests that those viruses are present in populations as viruses, but actually they're there as part of bacterial information. So we go ahead and do these analyses with these limitations in mind, and we produce images like this. So this is a chronochart, and when you're reading a chronochart, you start from the inside and you work your way out. So in the middle there are viruses generally, and then along the edge of the circle there's a more highly resolved idea of which viral species you're getting in your data set. So out of those 9 metaviromes and 15 million reads, this chronochart represents 6.9% of that. If you're feeling sympathetic and thinking, Siv, that's really depressing, I'm really sorry, thank you for being sorry. But really that's kind of what we expect for this kind of analysis. But the first thing that sort of jumped out at us when we were looking at this is it doesn't really look like there's that much going on. It's not really that diverse, which is sort of unsettling. Something else that kind of unsettled us was this. Right at the top of this chart, it's telling us that 4% of the whole 6.9% is represented by one phage, which is a Planktothrix phage. I don't know if it's because I'm British, but I'm inherently suspicious of everything. So what we did is we looked at the genome of this Planktothrix phage specifically and went back to hunt for it. So we sort of worked backwards a little bit. So on these charts, what you can see is that the x-axis represents the genome of the phage in question. The y-axis is showing you the number of hits to a specific area of that genome, and I think you can see straight away what's happened. There is no real distribution of hits across the entire genome. They're all coming back for this one ABC transporter, and that suggests that yeah, the ABC transporter is definitely there, but the phage probably isn't, which is kind of sad in the face of this chart. So as a fiddler, went back and looked at some of the other genomes that were popping up on the corona chart, and it's not all doom and gloom. For a lot of the other viruses, there's a much better distribution, but it still concerned us in as much as this is not entirely representative of what's going in our samples, or not as representative as it could be. So what we really wanted to do was focus on a specific group of phages and try and figure out the goodest way of getting a representative idea of the position that that group holds within these data sets. So a candidate for this analysis popped up pretty soon thereafter. This is PB1. PB1 is the type strain of the Pabuna-like viruses, and they've been found previously in lots of other places, freshwater soils and activated sludge. As a group, they're mostly Pseudomonas infecting, except for one, which is a Bacterial phage. PB1 comes in at around 65,000 base pairs. He's kind of a middling-sized phage. And in a collection of metaviromes that we were analysing in 2015, we were finding really frequent hits to PB1. So many that we began to wonder if we could construct a kind of Frankenstein-type entire PB1 genome from all the scraps of PB1 that were floating around the metagenomic dataset. And this has been done quite frequently in the last sort of 6 months to a year with bacteria and other viruses and metagenomic datasets, because everyone's producing so much data, right? So it's getting easier to do. So we wanted to try and piece together our 1PB1, and actually what we ended up doing is piecing together 9 new PB1-like genomes with really good coverage. Most of this was done by Emily down there. I think Emily's watching now. Hi, Emily. She looks really happy because she's got ice cream. So why wouldn't she be happy? Anyway, the figure in the top left-hand side of the screen is a comparison of the genomes of our Lake Michigan viruses with the existing Pabuna-like viruses. So the existing guys, they're all labelled in black. PB1's about halfway down. The blue names are viruses that we physically isolated from Lake Michigan and sequenced, and the red names are the phage genomes that we've sort of harvested from the metagenomic data set. So I think hopefully you can see that all of the Lake Michigan viruses are quite similar to PB1 and relatively dissimilar in comparison to the other Pabuna-like viruses. So while they are all very similar, they're definitely different. And the figure on the right-hand side of the screen is showing you the PB1 genome in the middle, in the center circle. Along the outside, the colored areas represent coding regions that are different in all the different phages. So we thought this was pretty cool because what it looks like is, for whatever reason, in this particular data set, these viruses are really abundant, genuinely abundant. And we didn't really know why. So the first thing that we tried to do was go back to other data sets and find them in there. So again, what we're seeing on these charts are the x-axis is the PB1 genome from 0 to 65,000, and the y-axis are hits to specific areas of that genome. Panel A is a collection of all the data that we've generated from Lake Michigan, all of our metaviromes. And panel B is a collection of lots of publicly available metagenomes. And this was kind of disappointing. Because what you can see is, again, there's not really an amazing distribution across the whole genome. And for Lake Michigan, we had these 2 big peaks, one for a structural protein and one for a DNA polymerase, and it's not really telling us much. And I was expecting to see something a little bit more consistent across the genome, but we didn't. And again, for the publicly available data sets, what we're finding is that the biggest peak is a hypothetical protein, one for a helicase, and not an amazing distribution. So what we did is went to these hits specifically, directly, and had a look at what was going on with them and what was being returned via our blast-based classification. And what was happening was this. We were getting really great E-values and really strong identity matches, but our coverage of our DNA sequences versus the ones already in the database were coming in at something like 40%, I think was the highest one. And for phages, that may as well be a match to Liza Minnelli or something. I don't know. It's not a genuine match. So what we wanted to try and do was figure out a way, a fiddly, creative, different kind of approach, specifically for the Pabuna-like viruses, to see if we could generate a more comprehensive, representative idea of where they stand in these data sets. And we ended up with this. I really like this figure, and it's really complicated, and I'm going to try and explain it in a really basic way. But we just had this paper rejected, and one of the comments that the reviewers made about this analysis is that it was awkward and unconventional, and that kind of really spoke to me, because that's sort of how I am as a person generally. And I think that in science there is a place for being an awkward and unconventional, because this figure works for us. And it came about as a result of lots of fiddling, a very intensive effort with regards to fiddling with this data. So, along the x-axis, or along the top, what you can see again is the PB1 genome from 0 to 65,000 base pairs. Along the y-axis are the labels for our metaviromes and publicly available metaviromes. The grey squares represent areas of the genome that we don't have any information for, and the coloured squares generate this kind of genome atlas of PB1. So the threshold for this analysis is the Burkholderia phage. Do you remember that I mentioned that there's one phage in the Pabuna-like viruses that infects Burkholderia? In order for a hit to get onto this graph, it needed to be a specific level of similarity, at the very least, to Burkholderia. So the blue squares represent more of a similarity to the Burkholderia phage, and the darker reddy orange coloured squares represent more of a similarity to PB1 specifically. Hopefully what you can see is that actually genuinely the Pabuna-like viruses are distributed quite widely across these data sets, but specifically in Lake Michigan. I have no explanation for why that may be, why they're so well distributed, and we put this figure together and submitted the paper, and I left. So we've still got a lot of work to do on this, but I think this is really interesting, and I think that this group of viruses potentially has a big role in the environment. So that's PB1, and I recently got some data back that suggests the PB1 strain that we're working with in the lab at the moment, which came from a culture catalogue, is pretty different to the current RefSeq record for PB1. And this is an aspect of working with phages which everybody knows about, which is a little bit tricksy. They change so frequently, and anything that changes at the genomic level could easily have an impact on the behaviour of the phages in the lab and therefore the environment. So there's a lot of work still to do with PB1, but I hope you'll agree that he's an interesting critter. So I'm going to talk a little bit about some of the stuff that I did for my first postdoc. This is a picture of the bloom that's happening or has been happening in Florida at the moment. It's taken from space. It's absolutely ginormous. I don't know how many of you will have interacted with cyanobacteria on an intimate level, but if you're walking your dog in the evening and you come across guacamole thick microcystis that's really going to ruin your day. I promise it will. For my first postdoc, I worked with cyanophages. These are phages that specifically infect cyanobacteria. And this was part of a really big EU project. And our role in this project was to try and figure out if we could use phages to remediate blooms in the environment. And I can tell you how that ended. The answer is no, you can't. Not at the moment anyway. But as a result of this project, I stumbled across a quite interesting virus. But cyanobacteria are really cool in their own right. They have a really ancient lineage. They're incredibly genomically plastic. They're photosynthetic. They're important in primary production of nitrogen fixation. They were responsible for oxygenating Earth's atmosphere. But when they proliferate in the form of blooms, it's really bad news. They bugger everything up. They're incredibly toxic. They produce neurotoxins and hepatotoxins. They generate gargantuan quantities of biomass, and they're positively buoyant, and they can regulate their own buoyancy and sort of hop around the water column. At the moment, we can disrupt the water column, and because these are exacerbated by eutrophication, we can try and limit the amount of phosphorus and nitrogen that's loaded onto controlled waterways. But really, because of climate change and because temperature makes this problem more problematic, we're transitioning to a phase now where we don't really know what to do about these blooms. And there are one or two really big ones that happen very reliably every year. So we're getting to the point where we're in trouble when it comes to cyanobacterial blooms. So as part of my postdoc, I generated a really big catalogue of freshwater cyanophages. I've got lots that I haven't looked at yet that I really need to. At the time, it was just me in the lab, so I went for the low-hanging fruit, and I picked the virus that was the easiest one to deal with. And these phages are not easy to deal with. I've got a whole slide where I'm just going to whine about that for a little while. But one of the most interesting ones, which was also luckily the low-hanging fruit, was MHI42. So from the AFM pictures and the TEM pictures on the right-hand side of the slide, you can see this is a podovirus. He has a viral capsid, and then he has a little stumpy foot for a tail. MHI42 has a broad host range and infects across a couple of genera of cyanobacteria. He's also a really big phage. So I guess usually for a podovirus, you'd expect a capsid diameter of around 60 nanometers, and MHI42 is kind of getting up to about 200. He also does weird things when he infects hosts, and demonstrates a switch in lifestyle in response to incubation conditions. So there might be a reason why he's so big. He has quite a large repertoire. MHI42 has got a big genome to go with that big capsid, and I have yet to sequence it. Unfortunately, there's kind of a quirk of some phages that they're just really difficult to sequence. But using PFG, I was able to determine that MHI42 is about 150 kb, which is kind of whopping. I just wanted to do a little bit of a plug because this phage was sort of the inspiration for Phage Fisher, which is a paper that we published this year that was headed up by Thomas, who's really amazing computational scientist, and he's going off to grad school next semester. And Phage Fisher is sort of this nifty little tool that you can use to separate anything from anything, really, but we used it to separate phage sequences from contaminating host sequences, bacterial sequences. So I just wanted to give a little plug for that and for Thomas. So regardless of the fact that this genome remains unsequenced, I did a lot of culture-based work with this phage. When you're working with phages at the bench, there are a number of standard methods that you can use to describe host range and growth kinetics and things like that. And most of them are based on plating. For these cultures, that's not really possible. They plaque inconsistently at best. They take a really long time to grow, the bacteria that is. And in the case of cultures like Planktothrix, they don't stay still on solid medium. Planktothrix glides and skids all over the surface of the plate, so you can't generate plaques anyway. I put in this picture because I think it's really cool. This is at the bottom right-hand corner of the screen. These are little mini cultures of Microcystis in multi-well plates. And these wells contain about 4 mils of medium. And you can see, hopefully I've highlighted it with arrows, the Microcystis forms such a stable layer at the top of these little mini cultures that when you apply phages, you get these little floating plaques at the top, which is completely useless and unhelpful, but I still think it's great, and I think it's a really nice demonstration of how these cyanobacteria can behave in the environment as well. That's a very stable layer of growth. So all of the work that I did with MHI42 was based on generating lysis profiles, and essentially what that means is you set up a liquid culture, monitor absorbance over time, and you expect to see a decrease in absorbance when the bacteria are killed through lysis. That's the most basic way of describing it. With MHI42, I, for the most part, worked with 4 different cultures, 2 strains of Microcystis aeruginosa and 2 species of Planktothrix. The Microcystis and Planktothrix are 2 really different hosts. Microcystis is unicellular, Planktothrix is filamentous. They're generally found in different areas of the water column. So it's interesting that this virus has the scope to infect these genera of cyanobacteria anyway. These graphs in the middle of the slide show you lysis profiles for the 2 Microcystis cultures. So along the x-axis, it's time in hours. These took a really long time to generate. The y-axis is a log scale of absorbance at 750 nanometers. So this is monitoring light scattering. The control in the top graph is a solid line, and you can see that it soars off gracefully into exponential growth. That's lovely. And then the dotted line represents an infected culture that's being lysed. And there's a crash there. The second strain of Microcystis aeruginosa, which is 1450/8, imaginatively named culture, demonstrates temperate growth, lysogenic growth. So instead of this dramatic crash, it just kind of keeps going. These are 2 strains of the same species, so ostensibly very similar cultures and 2 very different reactions after being infected by this virus. This virus destroys Planktothrix very, very quickly. I didn't include the graphs because the host doesn't even make it into exponential growth really before the virus kills it. So again, that's kind of a different reaction. I'm not sure why that is. It might be something to do with the morphology of the host. But again, that's something I need to look at in greater detail. The little graph on the right-hand side of the slide demonstrates something that happens with all of these cultures after you infect them. Before they die, there's an increase in growth rate. So this is a pretty obvious advantage for the phage, and it happens with all of them. Similarly, this is a graph of lysis profiles on the second strain of Microcystis aeruginosa, the one that originally demonstrated temperate infection. When you change the incubation temperature for the culture after it's been infected by a phage, you can see that there's a switch from lysogenic growth to lytic growth. This is unpublished data. I need to work a bit more on this. But again, it demonstrates that this virus has quite a wide repertoire when it comes to these cyanobacteria, and this is sort of the basis really for what I want to do over the next couple of years at Tech. I'm really interested in how infection affects these sort of 3 pillars of bloom formation. We've already seen with regard to biomass that the phage can increase growth rate. So what does that do on a larger scale? That's what happens in the lab, but what is it likely to do in Lake Erie or somewhere similar? How does infection affect toxin production? The chances are it infects it somehow, but it would be good to get a handle on exactly what. And again, with buoyancy, I'm not sure if it's likely to have an effect, but it will be really interesting if it does have an effect. And with a phage that has so far got an unsequenceable genome, I think there's lots more to discover. So that's really kind of the sciencey bit of my talk. I want to do a little bit of cheerleading for what's going on in New Mexico at the moment. For anybody who doesn't know, that's where New Mexico is. Depending on your perspective, being sandwiched between Arizona and Texas is a pretty nice place to be. I don't know how you feel about that, but I really like it here. At the moment, I'm in northern New Mexico, up in Los Alamos with LANL and the New Mexican Consortium. A little bit further south is Albuquerque, and that's where the University of New Mexico is. A little bit further south yet is Socorro, and that's where New Mexico Tech is, and then down in southern New Mexico in Las Cruces, that's where New Mexico State is. The New Mexico Consortium is a non-profit corporation that was formed by UNM, Tech, and New Mexico State. And it's pretty special. The idea is that these colleges really want to integrate with industry in the state. New Mexico is not a rich state, so anything that's good for academia and anything that's good for industry is good for everyone. So this is part of my job description while I'm here. I'm really trying to set up collaborations between the locals and between the academic institutions, but also one of the reasons that New Mexico is really interesting is it's absolutely stuffed to bursting with government labs who do lots of exciting top secret things. And New Mexico Consortium really aims to increase the role of Los Alamos National Lab in science, science education, and economic development generally in New Mexico. So part of the reason why I'm really excited about this aspect of my job is the facilities up here. I've worked in industry for a while and I've been in academia for a really long time, and I have never seen labs like these. I did my degree and my PhD at a small university in the UK, and I've got a working class background and blah, 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 blah. And being able to bring students, especially students who wouldn't get the opportunity to do this otherwise, into labs like these is amazing. So, for example, one of the most exciting facilities that we've got at NMC from my perspective is this setup. This is a room full of 35 miniature photobioreactors. They act as microcosms, as little mini lakes. They're monitored constantly by computers, and you can assess lots and lots and lots of different variables, and they're completely automated and they're absolutely beautiful. And NMC has money to bring students up here to use this technology and to set up their own experiments. So this is one area that I'm excited about because it has obvious use for the cyanobacterial work. In addition, NMC has lots of facilities for advanced computing, for modeling and analysis, and for biomedical technology. It is a remarkable facility, and anybody who's interested in any of these aspects, shoot me an email or go and check the website, because we're all about the more the merrier. We'd like to get as many people involved as possible, particularly undergraduate students and graduate students. So that's my little plug, and that's kind of also the end of the presentation. I probably zoomed through that really quickly. I have to say thank you to all of these people who have helped me get to where I am now, which is actually a really exciting stage of my career. So in particular, I want to say thank you to Catherine and Joy and Paul and Vanessa, who was the NHS microbiologist who taught me what bacillus smells like. And I have to say thank you to all of the people who've given me money at some point as well, because that's always nice, isn't it? Thank you for listening, and I guess if anybody has any questions, I'll take them.